Guozman Science. I'm your host, Mr. Gwanderson, and today we will be talking about a very important process in biology, and that is cellular reproduction. Okay, so in humans and organisms that reproduce sexually, there are two types of cells, somatic cells and gametes, which are the sex cells like sperm and eggs. And then the somatic cells are all our other body cells, like hair, skin, nails, eyes, I don't know, whatever you want. Everything but sperm and eggs. In somatic cells, somatic cell division occurs in a process called mitosis. Gamete cell division occurs in a process called meiosis. First, we're going to look into the cell cycle for somatic cells, and it consists of two parts. About 10% of the cell cycle is spent in mitosis, and mitosis consists of two parts, actual mitosis and cytokinesis. The other part of the cell cycle is interphase, and this is about 90% of the cell cycle. Interphase consists of three different parts: the G1, the S phase, and the G2 phase. The cell cycle allows for the distribution of identical genetic material called DNA to two daughter cells for somatic cells. Now let's get further into interphase. Interphase, like I said, consists of three parts. So let's look at what happens in the cell during those three parts. So during the G1 phase, the cell grows. And in the cell, we have DNA and these long strands that we call chromatin. Another structure that we're going to highlight in this very basic cell diagram, like I'm missing a ton of stuff, is called a centrosome. And it consists of two parts, so these two perpendicular structures that are called centrioles. And they're each centriole is composed of a bundle of microtubules, and microtubules are a type of motor protein, and you'll see what I mean by that. They move and they cause things to move. You'll see what I mean by that as we get further into actual mitosis, and they do a lot more movement. So during the S phase, the DNA in the cell is duplicated. So we have, let's just pretend that's double the amount. Super important step. Then during the G2 phase, the nuclear envelope begins to form around all this duplicated chromatin, and the nucleolus appears, and the centrosomes duplicate. This, kind of with the complete nucleus, because the nucleus will be formed by then, is what our cell looks like going into mitosis. The first part of mitosis, like I said, or the mitotic cycle, is mitosis. The second part is cytokinesis. And the 5 steps in mitosis are prophase, prometaphase, metaphase, anaphase, and telekinesis. For some reason I say telekinesis instead of telophase a lot in this video, so every time I say telekinesis, I meant to say telophase. Okay. So let's take a look at what is happening in the cell during each of these steps. So the first step, interphase. Again, I'm like really tired. I meant to say prophase, not interphase. We already discussed that. Okay, yeah. We have our cell membrane, and the chromatin that had recently duplicated and been enveloped in a nuclear envelope begins to condense, and the two corresponding duplicate parts are called sister chromatid, and so they condense inside the nucleus, and they attach in the center at a unit called a centromere. So our centromere is in the middle of each of these chromosomes that are the condensed chromatin from the previous step. The two centrosomes, the microtubules, begin to extend. And there's two different types. We'll get into what these two different types are once it makes a little more sense with what else is going on. These begin to extend, and these are the beginnings of the meiotic spindles, the microtubules extending. And as those microtubules extend, we move into prometaphase. During prometaphase, the centrosomes begin to really be pushed apart because these microtubules are extended. And how are they doing this? Like there's a nucleus in the way. Well, during prometaphase, the nucleus begins to break down. So here's our nucleus. And the chromosomes inside begin to condense even more. And if you'll notice, I'm drawing these yellow ones, so they're kind of starting to interact with the middle of these chromosomes. That is because, say here's our chromosome. We got our centromere right here. At either side, at both sides of the centromere, there's these proteins called kinetochores. And certain microtubules, kinetochore microtubules, interact with these and begin to kind of bond with them. Other microtubules don't. Again, during a later step, we'll see specifically what these guys are doing. During the next step, however, the centrosomes have completely pushed each other away. This next step is metaphase. And the chromosomes align at the metaphase plane, which is equidistant from both of the poles of the cells, because both the kinetochore interacting microtubules interact. They extend equidistance to the middle of the cell, kind of pushing the chromosomes in. And then, of course, we have these other microtubules. Our nucleus is completely gone. And these structures of these extended microtubules are known as the meiotic spindle. So our chromosomes are aligned in the middle of the cell. They're interacting with the kinetochore microtubules, and the centrosomes are at opposite poles of the cell. During the next step, anaphase, because of the non-kinetochore microtubules pushing each other apart, the cell kind of extends. So now it's kind of more of an oblong shape, because these non-kinetochore microtubules, like I said, they're pushing the cell apart. The kinetochore interacting microtubules, however, begin to shorten. And by doing so, because they're attached to the kinetochore at their corresponding side, they begin to shorten, and by doing so, they split that middle section, the centromere, in half, and so they're pulling apart the two sister chromatids. So now we have our daughter chromosomes. Finally, so these continue to shorten and shorten. During telekinesis, during telophase, two daughter nuclei have formed. So our cell is even more extended. And we have our nucleus forming. So there's two daughter nuclei. And our centrosomes, all the microtubules have kind of broken down. they're at opposite poles of the cell. And the corresponding DNA that they pulled apart begins to decondense and be enveloped in the two identical daughter nuclei that form. And the second part of the meiotic cycle, and the last part, is cytokinesis. And this is when a cleavage furrow forms, so I'll draw that here, between these two cells, causing them to split into two identical daughter cells. So by this point, our nucleolus has reappeared, our nuclear envelope is full and intact. In those centrosomes, we have one in each daughter cell, and that is mitosis.